Hello everyone, this is LATHA AKULA. In this video, we are going to study about mechanism of nitrogen fixation. So in the last video, we have studied about how the root nodules were formed. In that, what we have studied, the root nodules consists of an enzyme nitrogenase and one pigment like hemoglobin was present. What is the enzyme? Nitrogenase. So this nitrogenase enzyme that fixes the atmospheric nitrogen. And what is the atmospheric nitrogen? It consists of two nitrogens, between them 3 covalent bonds were present, right? So this nitrogenase enzyme, it is made up of molybdenum iron protein. What are the components present in the nitrogenase? Molybdenum and iron were present. And here, the molecule of nitrogen is reduced by the addition of hydrogen atoms. So how the nitrogen is reduced to ammonia? It is reduced by the addition of hydrogen atoms. And where the nitrogen atoms are coming from? They are obtained from reducing agents like NADPH2 and FMNH2. So this is the nitrogenase enzyme. And this is the substrate. What is the substrate here? Nitrogen gas is the substrate. The substrate nitrogen gas binds to the active site of the enzyme nitrogenase. See, it has bind, right? So here the nitrogen gas, it has 3 covalent bonds. So after binding, it was reduced by the addition of 2 protons. So here the addition of the protons, the nitrogen, it converted into diamide. Diamide means what? N2H2. 3 covalent bonds will becomes into 2 covalent bonds. And one more time it is reduced with 2 protons. And the second time this diamide is converted into hydrogen. Hydrogen means what? N2H4. How many hydrogens are present? 4 hydrogens, 2 nitrogens. This is called as hydrogen. And here the 2 covalent bonds will become into 1 covalent bond. And again one more time the protons were added and it gets reduced and there is no covalent bond. Finally there is no covalent bonds between the nitrogens and the hydrogen will becomes into ammonia. And how many ammonia are produced from the one molecule of the nitrogen? 2 ammonia. This is NH3 and this is NH3. Right? That means from one molecule of the nitrogen, there are 2 ammonium molecules are produced. See here, 2 ammonium molecules was released. And then this is substrate, it is free now. This free nitrogenase can bind to the another molecule of the nitrogen. First the nitrogen converted into diamide and then into the hydrogen and finally into 2 molecules of ammonia. And here for the synthesis of this ammonia, it requires high amount of energy. How much amount of energy? For 1 molecule of the ammonia, 8 ATP molecules are required. That means from 1 nitrogen molecules, how many ammonia were produced? 2 ammonia, right? So for 2 ammonia molecules, how many ATP are required? 16 ATP. For 1 ammonia molecule, 8 ATP are required. For 2 ammonium molecules, 16 ATP were required. And how this energy is obtained? This energy is coming from the respiration of the host cells. During the respiration process, what is released? Energy is released, right? The energy was stored in the form of ATP. That ATP energy is used for the reduction of this nitrogen into ammonia. So this equation is important here. From 1 molecule of the nitrogen, 2 ammonium were released. For the 2 ammonium molecules, it requires 16 ATP energy. And here, what is the enzyme required for the nitrogen fixation was nitrogenase. And this nitrogenase is highly sensitive to oxygen. That means this nitrogenase is active only under anaerobic condition. If there is aerobic condition, this nitrogenase will become inactive. Already the root nodules have adopted themselves to preventing the oxygen. If accidentally oxygen enters inside the nodules, there is one more thing is present, that is leg hemoglobin. This is a pigment, right? This leg hemoglobin will act as oxygen scavenger. What is the meaning of scavenger? It removes the oxygen. It protects the nitrogenase from the oxygen by removing the oxygen. Who is the oxygen scavenger in the root nodule? Leg hemoglobin. What is the enzyme required for the fixation of the nitrogen? Nitrogenase. So presence of this leg hemoglobin in the root nodules, the cells of the root nodules will appear red or pink color because of the presence of the pigment like hemoglobin. Like hemoglobin means what? We know the hemoglobin present in the blood. Because of the presence of that hemoglobin, our blood is red in color. But here in the leguminous root nodules, the pigment is present, right? So hence it is called as leg hemoglobin. This is about the mechanism of nitrogen fixation. Now we'll see about another concept, that is the fate of ammonia. So what is the product of the nitrogen fixation? Ammonia, right? So this ammonia, which is produced after the nitrogen fixation, will not remain in gaseous form in the soil. So it has to be, and the ammonium is the gaseous form. It has to be protonated to become the ammonium ion. So one proton is added to the ammonia and it will become the ammonium ion. So most of the plants, they can assimilate the nitrates and ammonium ions. But this ammonium ion is highly toxic to the plants. Hence, this ammonium ions has to be assimilated as soon as they have been formed. And these ammonium ions are assimilated in two ways. One is by the reductive amination process, and second one is called as a transamination process. So here the reductive amination means what? This ammonium ion combines with alpha ketoglutaric acid with the help of the enzyme glutamate dehydrogenase in the presence of coenzyme NADPH. So now this ammonium ion will becomes into the glutamic acid or glutamate. So the first acid is produced, right? That is the glutamic acid. So what will happen? The ammonium ion combined with the alpha ketoglutaric acid in the presence of enzyme glutamate dehydrogenase with the help of the coenzyme NADPH and produces glutamate or glutamic acid. And the next process is the transamination. The next step is the transamination. Transamination means what? Transfer of amino group. Transamination means transfer of amino group from the one amino acid to the keto group of the keto acid. That is called as a transamination process. And this transamination is mediated by an enzyme called as transaminase. The name itself indicates it is transaminase, or it is also called as aminotransferase. Here in this transamination process, what will happen? One amino acid is already produced in the first step, right? So that amino acid will act as a donor. And there is one keto acid is present which will act as the donor. This keto acid, it consists of keto group. C double bond O is a keto group. And here from this donor, this amino group is transferred to this keto acid. And then so what will happen here? Keto acid will become into the amino acid, right? See this? Here this amino group is transferred here and keto group is transferred here. And finally this is the reaction. And this reaction is mediated by an enzyme transaminase. So what happened here? This is the glutamic acid is with the oxaloacetic acid in the presence of transaminase. This oxaloacetic acid is the keto acid which consists of keto group. And then after the reaction, this glutamic acid is converted into alpha ketoglutaric acid by losing the amino group. And this keto acid, oxaloacetic acid, is converted into the aspartic acid by receiving the amino group. Then another amino acid is formed. Okay, so glutamic acid is a primary amino acid from which other amino acids are formed by the process of transamination. And the last concept in the mineral nutrition was amides. Here amides means what? These amides are nothing but these are the organic compounds. Amides are the organic compounds which are derived from the amino acids. Where they came from? They are derived from the amino acids. So in this one, the hydroxyl compound, if we see this is the amino acid, what the amino acid consists of? It consists of amino group, carboxylic group, and R side chain is present. And here in the carboxylic group, which component is replaced with another amino group? Then it will become amide. Amide consists of two amino groups. Amide consists of two amino groups. Actually, amino acid consists of only one amino group, but these amides, they consist of they were replaced with the hydroxyl group is replaced with one more amino group. Then it will become amide. So here So two amino groups are present in the amides. There are two important amides are present. One is aspargine and another one is the glutamine. Glutamine and aspargine are the amides. We need to remember glutamine and aspargine are the amides. And how these amides are formed? This aspargine amide is formed by the amidation of aspartic acid. So to the aspartic acid, one more amino group was added by replacing the hydroxyl group. Then this aspartic acid is converted into the aspargine, and which is having two amino groups. And the glutamic acid is also added with one more amino group, and it will become into an amide glutamine. So these are the amides which are obtained from the amino acid by the addition of one more amino group. And this, what is the function of these amides? The function of these amides was they perform two functions. One is the storage of excess nitrogen. The excess nitrogen stored in the form of amides, and also they transport the nitrogen to the different parts. And these amides, they contain more nitrogen when compared to the amino acids. These amides consist of more nitrogen because in the amino acids only one amino group is present, but the amide consists of two amino groups. Hence, these amides have a more amount of nitrogen. More amount of nitrogen is present in the amides, and these amides are transported to the other parts of the plants by the xylem vessels. How they were transported? Xylem vessels. And if you see in the soybean, the root nodules of the soybeans, they also export the nitrogen in the form of ureates. These ureates are also the nitrogenous compounds like the amides. They also have the high nitrogen content like the amides. So this is the important example. Soybeans, the nitrogen is fixed and exported in the form of ureates. And this is about the amides concept. With this we have completed the chapter mineral nutrition, and after this I will start with the reproduction in the plants, sexual reproduction in the plants.